Okay, so we'll start to a new topic, the Alzheimer's disease. This is the most common neurodegenerative disease, actually. And it's getting more and more attention because people, their lifespan actually is getting longer. And the disease, the most important risk factor is the age. Okay, so it's when people get older, their chance of getting this disease is higher. And there are also some of the family history in which that those usually, if they have family history, their onset will be earlier actually. So we watched the video last time, and then we're going to talk about some of the characteristics of the disease and possible mechanism so far that we learned from the study in this area. So and then you from after this, then you will get some idea about so the disease actually gives us the idea about how the brain functions, and also the importance of the learning and memory. And the memory actually is really we believe it is stored in the brain. So some of the major clinical features in the Alzheimer's disease. Now, the onset is incidence. So if you still remember the video, it's not just in the onset, it's not just the memory loss, but associated with other characteristics. For example, the patient will less likely to expose himself or herself to new things, and then their mood might be changing, and so on. So those are some of the more characteristics very difficult to judge. So that's particular in the older patients, because that when people get old, they might have, the common, when people get old, you get forgetful and so on, forget the key and so on. Now, the one that actually people will start to recognize is the memory loss. Okay. Usually is the formation of the new memory, for example, forget the key, forget cooking and so on. Those are the initial, the most easy to recognize. Okay. Now those doctors, sometimes you talk to those patients and then ask them something, and then maybe present them with something. For example, you talk. The doctor may suggest, today it's a beautiful day, and then he had, for example, he did something in the morning, and then after a while, talking a little bit, about the news a little bit, for example, and then you ask the patient again. And then the patient usually, if they are starting to be affected by the disease, they will not remember. Okay, those are some of the initial, features of those memory loss. As I mentioned, the language impairments as well. So language is a very important, useful measurement, or to look into the person's the brain functions actually. Yes, particular if you look at the grammar or the words and so on. Now the other one is the visual spatial dysfunctions. If you still remember the video in which that that actually he is an actor mentioned about that. He's that actually upside down the menu, so those are different visual spatial dysfunctions. Okay. So memory actually composed of at least two different processes. So the learning and storing the new information, and then recall of the periods that acquired the information. Now, dementia first affects the process of the learning and store of the new information. So that's the reason why that those patients usually they will avoid learning new things and so on, and also will be forgetful. So those are the formation of memory. Now, you might see that, I put dementia here. Dementia actually is forgetful, okay, particularly in old people. Now, dementia is not just, not all dementias are Alzheimer's disease. Alzheimer's disease are one category of dementia that actually are associated with some of the pathological characteristics that we will talk about, okay? So not all the dementia are Alzheimer's disease, okay? There are different kind of dementia, okay? So in the Alzheimer's disease, this is continuous. So what is it called by neurodegenerative? Means that actually it will be progressive, getting worse and worse. And sadly, there's no treatment so far can slow down the process. Okay, so it's and the symptoms will get worse and worse. Okay, in those the disease actually in the process. So initially the formation, learning and storing of the new information, but eventually even the remote past becomes impossible to recall. So it cannot recall old memory. So you should still remember in two of the videos that mentioned about they cannot even recognize their partners or their wife, husband and wife, they cannot even recognize, or even the daughter at the last video. So those are the formed information stored in the cortex, cerebrum, actually, that disappear as well. So the learning deficit of those patients actually is similar to amnesia that caused by focal damage to the hippocampus or the diencephalon. Okay, so that means that for some reasons that there's a focal damage, in stroke or in other accidents that cause the amnesia in those patients. Those are actually similar to those dementia patients as well. So that means that correlated, there's actual changes in the structure of those Alzheimer's disease that actually cause the effect. So language, as we mentioned, is involve a very comprehensive use of the speech and the ability to read and write. So it is a complex process. So for example, if you ask you to, generate a sentence, then that involves the syntax and the words, and then when you speak out, so that they have to be in grammatically correct and so on, So in those Alzheimer's disease patients, they will show different deficits in the aspect of the language. So including the structure and the meaning, okay? So it kind of cannot comprehend, fluent in the language, because like for us, I mean, if you still remember or you have seen some of the children or those toddlers when they're really young, or I don't think that you will remember how you learn the language. You talk about, this is a car, this is an orange, this is a banana, and so on. And then slowly you form that the sentence. For example, I eat a banana, and so on. So those, syntax actually will need you to think of it. So if I say I eat a banana, then you at least that you'll think about a banana which is in yellow color, and so on. And those are learning curve, learning slowly. Now in the Alzheimer's disease case, then that actually will slowly deteriorate. just like going back. Visual and spatial functions, another very good, very important indications of those. The positions of the things, and then whether you can navigate the system. And that's the reason if you sometimes you heard about some old people with Alzheimer's disease in the news that actually they got lost in different places because the visual and spatial input actually is affected. Okay. Now there are tests that actually in the for doctors to identify patients. Now we will talk about actually how to identify those patients. Actually, so far it is actually not an easy task to identify or test those patients. There are ways to test that, but we will talk about those actually. So, for example, the test actually is very used to, for example, copy simple drawings to differentiate left, right, top, and bottom. Okay. Particularly, so we will look at the pictures later. Is a drawing of a clock. Now, actually, it is actually another way to diagnose how serious is the patients at the disease level. Okay, if they are performing poorly in the test, actually they will have a higher mortality rate because that also indicate how serious or the progressive of the disease. So, for example, among low scholars, 26% will die actually after several years, even though they are younger. So, if they have a score worse in the visual spatial diagnosis, then actually they will have a higher mortality rate, which is also linked to the degree of the seriousness of the disease. So this is one of the things that you can ask the patients to copy this clock with the numbers and with the pointers. And you see that, some of those actually are very bad in the actions. Some of those are much better. Some cannot even, put in the numbers. Okay. So you might see that actually this might be something you learn in your primary school, drawing the clock, the time. And so this is just the example of how to test the visual and spatial ability of those patients.
This is what they see.
No, actually, this is what, so they asked the patients to copy this picture. So they copy and then they draw those over here. We can see that this one is still okay, but those are one actually you can see that some of them they even cannot, because this is a very important test. So you have to look at the picture, and then you have to distinguish left and right, top and bottom. Okay, so as you can see that they cannot do it properly. So it depends on which patients, because you have to look at it, remember, and also, so this is testing your eyes and spatial, and also your hand movement as well. Okay, so you can see, you should still remember the video. Some of those patients, particularly the last two that show, they cannot move properly. They cannot, doing some daily tasks. Can the doctor determine how severe the patient is from the drawing? So, for example, if the drawing is like this, then the patient is supposed to be quite serious, okay? Or this one. Yes, they can have an idea. So there's a scale. We will talk about, for example, they will determine whether it's stage 1, stage 2, or stage 3, okay? But this is not absolute diagnosis still. So this is what I'll talk about. There are progressive changes in 2 or more areas of cognitions. One is memory, the other is maybe including language, mathematical ability, praxis, abstractions, and visual spatial perceptions. So, for example, in the first stage, there will be a memory impairment, disorientation in time and place. So you see that, those patients that, you heard about in the news that went for like a morning walk, but then wasn't able to get back to home. Those are one you see disorientation in time and place as well. Restlessness and anxiety, then this is secondary, maybe secondary because he cannot remember things. Then it will become anxious. There's some time that people will start to see whether the patient needs to go to see the doctor. Now, the second stage will be much more serious. So dysphasia is actually cannot generate ability to in the speech talk about something and so on. Apraxia is actually cannot move properly, doing some things. Anoxia is actually cannot properly sense things, receive information from the outside. Now, second stage will be more serious, blunting of the emotions and apathy, and also neurological signs as well. Some of the more impairment cannot move properly, or sometimes they will be, depressed. Now, the third stage is the last one. There will be a global cognitive decline. Cannot, respond to outside maybe. Loss of personality and incontinence. Incontinence actually cannot, control their body at all. So it's need the full actually care. Now the disease is actually very destructive to the family because that also require a lot of resources to take care of the patients. So that's the reason why that actually it is taking so much attention. How long will it take to enter the next stage? It depends. So all the patients will depends on their progress, but some may be faster, some may be slower. Depends on whether it is caused by family history or inherited or the individual. But it will not be a short term, as in the video, some of those like say 5 years and then they pass away. But getting to the second stage and then they will move faster and faster. If you have watched the news about Charles Koo, you can see, that the time that he was getting the Nobel Prize, still okay. After a while, they have done, interview with him again, and then if you have watched those documentary, and you will see that later stage he would just stay there and not moving at all. So that staging depends that it depends on the patient for how long it will last. So the clinical criteria proposed for AD are, first of all, it is incidence onset, so it's not very clear cut. It's not just that you have a cold and then it's boom, then you have the sore throat and so on. So it's progressive and slow at the beginning. The dementia occurs in the absence of other systematic or brain diseases. So it's somehow the memory formations will appear because that might be the most obvious, things that you can see. But actually, there might be other, for example, the personality may be changing a little bit and then become upset and so on. There's no absolute diagnosis except in the final stage is the neuropathological examinations. Okay, so the reason behind we'll look at it because there's a characteristic pathology in Alzheimer's disease, and those will be different from other dementia. Okay, some of those now actually have the psychiatrists or neurologists that can do the psychological examinations. If that is together with the CT or MRI or PET scanning imaging, that might give the doctor 80 to 90% accuracy. But still, the exact 100% diagnosis require the neuropathological examinations. So that's the reason why that actually, for example, in Hong Kong, how many people got the Alzheimer's disease actually is approximate, because usually when those patients die, most likely the family will not do the neuropathological examinations, because that requires to remove the brain out, cut it into pieces. And as you know, that in terms of Chinese culture, that is not a very common things to do for the patients to do, for example, to donate the brain for examination. So there are some of the adoption of the categories, for example, definite, probable, and possible, as by the task force on the Alzheimer's disease. So a diagnosis of the definite, actually, that will require the histopathological study, because there are characteristics of those in Alzheimer's disease, okay? Which is actually, just to let you know that the doctor that first described the disease is Alzheimer's. So that's the reason why it is called the Alzheimer's disease, the Dr. Alzheimer. So the probable AD can be diagnosed if there is a typical incidence onset with progression. So for example, follow those stages, stage 1, stage 2, stage 3. For example, if it is stage 2, and with the history, that's a more probable. Now, another one is possible. So if the presentation is somewhat like losing the memory, but there is no other associated conditions. For example, there's no head injury and so on. It's just memory loss. That will be possible. So you can see that actually it might be in the first stage or progressive in the second stage. Okay. So this is the brain image. So this is the brain take out and then cut it into slices. So most of it is the coronal sections. So this one is the control, normal individual. Now this is the stage 1, and this is stage 2, and this is stage 3. So you can see that actually there's progressive loss of the tissues. You can see that is the space is getting bigger and bigger. And particular, for example, here is the hippocampus. So you can see that here is still a little bit. So this one is normal. Then actually the part will shrinken, and you can see that actually the cortex will be shrinking as well. So that's obvious. That's a actual loss in the tissue. Okay. And you can see that actually the ventricle will enlarge because the loss of the tissue, and the space is occupied by more CSF fluid. Okay. So this is also tells you that actually the tissue loss is actually associated with the memory loss. Okay. So what is the characteristic of those? Now, one of the particular important is the amyloid plaques. Okay. those are the initial study. So in the initial study, so how to study the disease. So take out the brain samples from those patients. And then at that time, they will do the so-called silver stain. What happens? Silver stain actually, so at a pathological study, usually you cut out the samples and then you stain it with different things and observe the characteristic of it. Now there are two characteristics. One is the amyloid plaques. So those are staining from the silver stain, and then there's a a plaque just like really tiny in the tissue. There's another one is neurofibular tangles that's inside the neurons. Okay. So those plaques over there, and also that will be the neurites will be also affected. So that's how initially the disease was characterized. So that's the reason why in all forms of dementia, if you find those patients with the amyloid plaques and the neurofibular tangles, that means that actually this is the Alzheimer's disease. Does AD only affect the brain or also affect other neuronal system? The most obvious one is actually in the brain. So, but it's difficult to tell actually whether it also affects other neuronal system. There's no report for they study the peripheral nervous system, but mainly they look at the plaques. But that would be interesting things to look at as well. But you can see that actually those patients, their physical movement is still, well, at initial stage. Their autonomic nervous system still working, But when it's getting the later stage, that will be difficult to distinguish whether it is the central nervous system or peripheral. Okay. So normal dementia does not produce plaques and fiber tangles. That is not necessarily the case actually. They will not have those obvious formation of the plaques. The plaques are actually the one that actually to prove whether this is Alzheimer's disease or not. But some other dementia, actually they don't have the plaques or tangles. Okay, but they have other pathology. Yeah. Okay. So this is the PET scan to look at, normal and the AD patients. Okay. Usually CT scan also will show that actually there is a severe atrophy, and you can see that actually usually the brain weight actually will reduce of 30 to 40%. Okay. This one is actually interesting. It's not at a specific area, but actually overall. But it starts from some space first and then progressed fully to the whole brain. So here is an example of the scan. So this is a normal one. This is a stage one. So you can see that actually some of those at the optical loop and also temporal loop. So that's the reason why that actually maybe so that affected visual, spatial area, and also temporal and frontal loop. So temporal loop actually controls some of the language ability. So at the stage 2 that we progress to different area. So some of the minor places that maybe the motor cortex and sensory cortex. And this is later stage and image that actually the whole brain actually is severely affected. And you can see that the activity also decreased, most likely because of reductions in the neuron. So there's an actual physical reduction in the neurons. And so that's the reason why that, we believe that actually the memory actually is stored in the neurons. And then when the neurons actually degenerate, that actually go with the neurons as well. Okay. Now the cause of the Alzheimer's disease. So the cause will give us some idea about what initiate the disease. Now it's around, you can see that around 77.5% actually are not known. Okay. Now some of those are related to head injuries. It may not be the same. For example, I talk about the chronic traumatic encephalopathy in those athletes, particularly the ones that got the concussion all the time. We will not go through that, but those patients also have the tangles and the plaques. Now, those in some of the cases, the older people will have those as well, but depends on how much or that it have enough effect on them. So some of the head injuries, patient will also have developed the Alzheimer's disease. Now, some of those actually are caused by familial, okay, with family history. Some of those actually are caused by autosomal dominant. We'll talk about some of the autosomal dominant. So that means that if it get the mutation, it will have the disease. Now, so far, there are cases that actually find out. So particular in the case of the so-called APP mutation. So why actually the finding of this actually prove that actually is the problem in some of the protein metabolism. Now, plaques are usually a spherical extracellular lesions around 5 to 150 micrometer in diameter. So certainly it is small, but you can observe under the microscope. They are usually deposit of an insoluble fibular form of beta amyloid, A beta, okay, beta amyloid or A beta. And sometimes it contains the ApoE and components of the complement cascade. So that means that there might be some inflammatory response around those regions. Okay. So it's surrounding its core. So there will be a dystrophic neurite. Those neurite, the neurites are swollen and damaged. There are microglia cell and astrocyte activated. So there's an inflammatory response around that region as well. So that's what people believe, that the plaque is the major reason behind the degeneration of those neurons. Okay. Now, in addition to that, there are diffuse plaques that are non-fibular beta amyloid deposit in some of those. Those are the diffuse one, that one without the dystrophic neurite. So why is this the case that some of those are more damaging than the others? So that is still not known. But that when we talk about the more mechanism, that actually you will have some more ideas. So those diffuse plaques, actually, they are regions that are not clinically implicated in the Alzheimer's disease. For example, the thalamus and cerebellum. So even in and some of them, actually, you can see that in the brains of the normal human. So that give idea that actually when people get old, they will also have some of the diffuse plaques as well. But that is not enough to affect. So whether it is first start with the diffuse plaque and then progress to the more the insoluble plaque, fibular plaque. So that also scientists are trying to determine. Okay. So So the connection between the familial one and the unknown cases actually are linked by this. So-called the APP, amyloid precursor protein. Okay, because actually this protein, after processing, will form the beta amyloid plaque. Okay. Now, the function of the APP amyloid precursor protein actually is still exact function is not known, but some of those suggest that it's a growth factor because that when the brain actually having injury, that actually will secrete, this APP out. Now, depends on how that is processed. It can process to A beta 40 or A beta 42. And actually, A beta 42 is more toxic to the neurons. Now, in the mutation, in those patients with the family history, they somehow their APP will process into more of this A beta 42. Okay. In cell or in neurons, actually, A beta 42 is more toxic to the neurons. Now, there are also argument or ideas whether it's the soluble one or the insoluble one actually is more toxic to the neurons or causing the neurons dysfunctions. That also has been argued, and there's still no argument, no legal conclusion. Okay, so anyway, that will form the aggregations that may form the amyloid plaque. So that believed to cause the neuron cell death. Now, the other pathway being affected is some of the kinase, phosphorylation of the tau. Now, the phosphorylation of the tau actually can induce the formation of the tangles inside the cell, inside the neuron. So that whether that is also contribute to the neuronal cell death. There are crosstalk between these two, whether it's the plaques or the neurofibular tangles, still under debate. Depend on which side of the scientists. So some of the scientists believe that tau is the problem. Some of those believe is that tangle is the problem, or the plaque is the problem. Okay.
That means, according to our current understanding, we only know the hallmarks, the pathological hallmarks, but not the details mechanism.
I will talk some of the detailed mechanism, but this is something that that has been studied for very long, and there's still no way to treat the disease. Even we know that. So, for example, some people will suggest that if you remove the A beta or reduce its productions, then that can be as a treatment. Okay. Some other in the tau case. Tau actually is a protein, is microtubule related. So when it phosphorylated, it will dissociate from the microtubule. And then somehow they will form paired helical filaments. And those has been also shown to be toxic as well. Okay. So so the beta amyloid, A beta, actually derived from the amyloid precursor protein. The protein is actually processed by protease. Okay. So the A beta peptides are normally found in normal human CSF as well, and it is processed by the protease in the membrane, sparked protease. So mutations in the APP actually give rise to actually around the cutting site. So the mutations that cause the Alzheimer's disease actually clustered around the processing site. So that's the reason why that we believe that that is the problem, and the processing of those actually will result into more formation of the A beta 42. So this one, A beta 42. So that's the reason why that scientists believe that that is the problem. Okay, so how it is being processed? So APP processed by two pathways, and only one of the pathway give rise to the A beta peptides with 40 or 42 amino acid. The A beta 42 is the one that actually more toxic than found in studies. So there's another pathways that actually will process the A beta into another peptide, and so that's the reason why the A beta peptides actually is the problem. So the first pathway actually is first the APP is cleaved by the alpha secretase. Okay, then there will be remaining the C-terminal fragments containing 83 amino acid. Sorry. So that is further cleaved by the gamma secretase. Okay, so become the P3. So this is not actually a problem for the case, but the processing in a more larger extent actually is by the second one. So in this one, beta secretase cleaved the APP, and then that will leave a so-called C-terminal component of 99 amino acid. That is further processed by the gamma secretase, okay, to become A beta peptide. And that one is the one that actually is problematic for the case, okay? So at once you see that actually the secretase actually may be a possible way to interfere and find the treatment. And also actually the secretase, if there's mutation as well, that actually will affect the development of the disease, which is the case actually. So what is the functions of APP? So APP protein may function as a protease inhibitors, or cell adhesion molecule, or neuroprotective agents against the glutamate excitotoxicity. So we are not quite sure yet. So still, so because a lot of the study actually is focusing on the pathological effects of it. Okay. Now the APP expression is increased during cell stress. For example, in the ischemia or head injury. So that's the reason why that actually, if those patients have stroke or if they have concussions, some of those actually they find that after that they would have plaques developed. And there are cases in which that after stroke, that will lead to the development of the Alzheimer's disease as well. Okay. Now the APP gene is in familiar early onset. So that also shows that it's important because early onset means that those patients actually would develop the disease much earlier. That means that they have, so that proves that actually the accumulation of those A beta is through a long time process. So that's the reason why the aging is one of the most important risk factors in the development of the disease, because it depends on the aging process. So enhanced beta secretase or alpha secretase, so that it also have the tendency also so that mutation increase the production and deposition of those beta amyloid in the plaques. Okay. Okay. So the hypothesis that beta amyloid contributes to the pathogenesis of the AD. So it's based on the assumption that the beta amyloid produced in excess, then will deposit, and then they will aggregate and form fibular plaques. Okay, and then the plaques will cause the neurons to die, and then progressively, after the neurons die, then that will affect the functions of the memory, and then later stage actually destroy all the memories. Okay. Now there's studies actually shown that the beta amyloid actually initially is a soluble, peptide, but then they may aggregate and then become insoluble, and that factors. So whether it is the aggregations or the pauses, actually, or also other cases that facilitate the aggregations of the beta amyloid, are actually considered important in the disease progressions. So what it is supposed, so it's very straightforward, seems to be, but still there are cases. So you have productions of the beta amyloid, depending on the individual, that will accumulate. If it's getting more and more, that will form the insoluble fibula, and that will form the plaques, and then that will, cause the neuron to degenerate. Now there is another component, actually, the ApoE epsilon-4 isoform. Actually, this one actually ApoE can enhance the aggregations, and in those cases, in the mice model, actually that can enhance the APP, the APP plaque formations. Okay, so in the animals that has been done for transgenic, so you put in the mutant APP form, then those mice will get the plaques formed. If it is in the ApoE, most mice that actually they're lacking the ApoE gene, the offspring will have less plaque formation. The ApoE actually is helped in the lipid metabolism. Okay, so that's the reason why that the lipid metabolism is believed also contributed to the disease. Okay. So how might abnormal amounts of beta amyloid lead to the Alzheimer's disease? It is that beta amyloid is actually very similar to the peptide transmitters. So are they also similar to peptide? It is still not known. In the cell culture, actually, beta amyloid is neurotrophic in low dose, but when it is high dose, then that become toxic. So when it is become helical filaments, that it become tangles as well. Okay, so that actively, so there's the beta amyloid actually in high concentrations can induce the formations of the paired helical filament, the tangles. Okay, so that's the reason why that there's a hypothesis that plaques actually induce the formation of the tau paired helical filaments, and then that cause the cell death. Okay. So just remind you, just Wednesday, that will be the project presentations, and we'll go to online mode for the next 3 classes. Okay. I will post an announcement later today or tomorrow to remind all of you guys. Okay. Stay online, and then we will first have will the present content included in the final? Certainly not. So I will not test you that many things. So the presented content will not be tested in the final exam. Final exam is non-cumulative. So the one that actually the midterm, I will not test you again. Well, let's go through the project first before you think about the final exam. Any things that you want to ask regarding the project? The first group, the number, from top to bottom, yes. Can you give you? No, I think the time is should be enough. Yes. Yeah. You have to try to make a good time arrangement. How many groups on Wednesday? We will try 3 groups first. So what happens should be 3, 4, 4. Okay. So the first day, let you have a more time to familiar with the setup.
I actually have a question because
Yes.
for my group, want to try out the remote control function of Zoom, but I'm not sure is it possible to do so if you don't have the access, like for the host to have that access. So can we try it out after the class today?
What do you mean by access?
Because for the remote control, like it's like I'm sharing the screen and then my groupmate can control my computer. But I think it needs the host to have that open that preference too. So like after the class, can I try out if the function is working in this class?
Yeah, sure.
Thank you.
Let me see. Yes, we can try. So the problem probably may be the share. I need to add you at the group present to be the co-host or something like that. Yeah, depends on. Okay, we can try. So you have your PowerPoint ready, or you just want to try?
I think we're nearly ready, but I just want to try, is it possible to do the remote control during the presentation?
What do you mean by remote control, actually?
It's like, because we have like several group mates, and then only one of us can share the screen, right? So we can open the function like I'm the one who is responsible to share the screen. And then my group mate can ask me requests to like control my computer so that they can control my computer and like change the slides themselves instead of me doing it for them.
But that one is actually not controlled by the Zoom.
That one is that function is from Zoom, but I think it may also needs the host open that preference in order to do so.
Oh, okay, I don't know about that function, but we can try actually. Any other students want to try, or? Let me see how far are we. Okay, so we will spend maybe 5 or 10 more minutes before, maybe 5 minutes. Let me finish these 2 slides, this one and the next one, then we can try it out. Okay. Okay. So you can see that actually the A beta that is processed by the presenilin or A mutations, age-related, that will affect the nervous system in different cases. There are different possible pathway, and believe that at the end, that actually increase the calcium, in which the calpain activated, and then there's a kinase, CDK5, actually can phospholipid tau induce, and then that actually become the new fibular tangles, and then that induce the apoptosis. So this is some of the pathways that believe that to involve. So this is similar to the excitotoxicity effects, because in the neurons that they find that there's a indications of the apoptosis in those Alzheimer's disease patients. Okay, so that's why the reason why that actually that involve those the calcium increase and then the tau phosphorylations and so on, because that mutation in the CDK5 kinase, if that increase the phosphorylation, actually that can induce Alzheimer's disease like phenotype in the mice. So that's the reason why that it has been studied like that. So that's also leads to the study that whether you can inhibit the apoptosis in the neurons that can prevent that from the neurodegeneration. But so far, there's nothing that has been shown to can affect the process. Okay, so maybe we will stop here, and then let your student to try. Okay, you can try now. So what do you want to do? So can I hear you? You mute yourself. Huh?
Oh, first of all, I need to share a screen first.
Okay, I will end.
I think you didn't allow us to share the screen.
Okay, let me see. Let me see. So I let you become the co-host, and then you can try.
Venus, are you here? Can you try to ask requests from me and see if it's working?
One guy doing it, the guy will die. I didn't think that was because I think it just because of my final year project. It's kind of related. Same reason why I never put that song.
My groupmate said they can't find the button. I think maybe it requires professor to open the setting too. So it's in my setting, and then there's a preference for remote control. Can you try to also open this function so that we can see if we can do it in this meeting?
Let me check. Okay. Maybe you can.
Stop sharing first.
No. Let me check whether I can do that. Okay.
Okay.
Okay. Okay. Okay. So, anyone, any questions? If not, that we'll end here. Okay. We will see on Wednesday. Okay. Okay. So we'll end here. Okay. See you on Wednesday. Okay. Bye bye.